Mitosis is one of the most important processes happening in your body right now. Understanding mitosis helps us understand how we grow, heal, and stay alive. Mitosis is a type of cell division that creates two daughter cells. Each daughter cell receives exactly the same number and kind of chromosomes as the original parent cell. Mitosis matters for three crucial reasons. First, it enables growth, allowing your body to get bigger from a tiny baby to your current size. Second, it enables repair. When you get a cut, mitosis creates new cells to heal the wound. Third, it enables replacement. Old cells wear out and die, so mitosis creates fresh new cells to take their place. Think of cells like building blocks. When one cell undergoes mitosis, it divides to create two identical building blocks. These blocks can then be used to build tissues and organs in your body. Here's a simple animation showing how mitosis works. We start with one parent cell containing chromosomes in its nucleus. During mitosis, the cell divides to create two daughter cells. Each daughter cell receives an identical copy of all the chromosomes from the parent cell. Mitosis is essential for life. It allows organisms to grow from a single cell into complex multicellular beings, repair damage, and maintain healthy tissues by replacing old cells with new ones. The cell cycle represents the complete life story of a single cell. It's not just about cell division, but encompasses everything a cell does from the moment it's born until it divides to create two new cells. The cell cycle consists of two major phases that work together in a continuous loop. Think of it as a circular journey that every cell must complete. The first and longest phase is called interphase. This green section represents the majority of a cell's life, typically about 90% of the entire cell cycle. The second phase is the mitotic phase, or M phase for short. This teal section is much smaller because cell division happens relatively quickly compared to the preparation time. During interphase, the cell grows larger, produces proteins, replicates its DNA, and prepares all the materials needed for division. This preparation phase is absolutely crucial for successful cell division. The M phase is when the actual cell division occurs. The cell divides its nucleus and then splits into two identical daughter cells, each ready to begin their own cell cycle journey. This continuous cycle ensures that cells can grow, repair tissues, and replace old or damaged cells throughout an organism's lifetime. The cell cycle is fundamental to all life on Earth. Understanding the cell cycle helps us appreciate how complex and well-organized cellular life really is. Every cell follows this precise timeline to ensure proper growth and division. Interphase is the longest phase of the cell cycle, where the cell spends most of its time growing and preparing for division. During this crucial period, the cell must double its contents and replicate its DNA before it can successfully divide into two daughter cells. Interphase consists of three sequential phases. G1 phase focuses on cell growth, S phase is when DNA replication occurs, and G2 phase involves further growth and preparation for mitosis. G1 phase, or GAP1, is the first growth phase. During this time, the cell increases in size, produces essential enzymes and proteins, and carries out its normal cellular functions. The cell also accumulates the materials and energy it will need for DNA replication. S phase, or synthesis phase, is when DNA replication occurs. Think of this process like making a copy of a recipe. Just as you would carefully copy each ingredient and instruction to create an identical recipe, the cell must precisely duplicate every piece of genetic information. Here we see the original DNA double helix. During S phase, this entire structure must be copied exactly to create two identical copies. First, the DNA double helix unzips, separating the two strands. Each strand will serve as a template for creating a new complementary strand. New complementary bases are added to each template strand, creating two identical copies of the original DNA. This process ensures that each daughter cell will receive exactly the same genetic information. G2 phase, or GAP2, is the final preparation phase before mitosis. The cell continues growing, produces proteins necessary for chromosome condensation, and duplicates organelles like mitochondria. The cell also performs quality checks on the replicated DNA to ensure accuracy before proceeding to division. By the end of interphase, the cell has doubled in size, replicated all of its DNA, and duplicated its organelles. The cell now contains everything needed to successfully divide into two identical daughter cells during mitosis. Mitosis is a carefully orchestrated process that occurs in four distinct phases. Each phase has a specific purpose and ensures that chromosomes are separated accurately between the two new cells. To help you remember the order of these phases, we use the acronym PMAT. This stands for prophase, metaphase, anaphase, and telophase. Each phase of mitosis has a specific role in ensuring accurate chromosome separation. In prophase, chromosomes condense and become visible under a microscope. In metaphase, all chromosomes line up perfectly at the center of the cell, forming what we call the metaphase plate. During anaphase, the sister chromatids separate and move to opposite sides of the cell, ensuring each new cell gets an identical copy of every chromosome. Finally, in telophase, new nuclear membranes form around each set of chromosomes, creating two separate nuclei within the cell. Remember the acronym PMAT to keep these phases in order. This four-phase process is crucial because it ensures that each new cell receives an exact copy of every chromosome, maintaining genetic accuracy. Prophase is the first and longest phase of mitosis, where the cell makes critical preparations for division. During this phase, three major events transform the cell's internal structure. The first major change is chromosome condensation. The loose chromatin threads that were scattered throughout the nucleus begin to coil and compact into visible, distinct chromosomes. This condensation makes the genetic material easier to move during cell division. Next, the nuclear envelope that surrounds the nucleus begins to fragment and dissolve. This breakdown allows the chromosomes to interact with the spindle apparatus that will soon form. The nuclear membrane essentially disappears, removing the barrier between the nucleus and cytoplasm. Finally, the spindle apparatus forms as the two centrosomes move to opposite poles of the cell. From each centrosome, spindle fibers extend throughout the cell, creating a network that will capture and move the chromosomes. These protein fibers are essential for ensuring each daughter cell receives the correct number of chromosomes. By the end of prophase, the cell has been completely reorganized. The chromosomes are now condensed and visible, the nuclear envelope has disappeared, and the spindle apparatus is in place. This careful preparation ensures that the genetic information stored in the chromosomes can be accurately distributed to the two daughter cells that will form. During metaphase, the cell reaches a critical checkpoint. All chromosomes must line up perfectly at the center of the cell before division can continue. The cell has already completed prophase. The nuclear envelope has dissolved, and the centrioles have moved to opposite poles of the cell. At the beginning of metaphase, the condensed chromosomes are scattered throughout the cell. Each chromosome consists of two sister chromatids joined at the centromere. The metaphase plate is an imaginary line that runs through the exact center of the cell. This is where all chromosomes must align before the cell can proceed to the next phase. Spindle fibers extend from each centriole and attach to special protein structures called kinetochores at each chromosome's centromere. Now the chromosomes begin to move toward the metaphase plate. The spindle fibers pull and guide each chromosome to align at the cell's equator. Let's examine the key structures more closely. This detailed view shows how everything is organized during metaphase. The centrioles anchor the spindle apparatus and ensure proper chromosome separation. Spindle fibers form the mitotic spindle that captures and moves chromosomes. All chromosomes must be perfectly aligned at the metaphase plate before proceeding. Each centromere has kinetochores that serve as attachment points for spindle fibers from both poles. Metaphase is a crucial checkpoint phase. The cell verifies that all chromosomes are properly attached to spindle fibers from both poles before allowing division to continue. This ensures each daughter cell will receive exactly the right number of chromosomes. Anaphase is the third phase of mitosis, where the most dramatic event occurs. The sister chromatids finally separate and begin moving to opposite ends of the cell. At the start of anaphase, the chromosomes are still lined up at the cell's center, just as they were during metaphase. Each chromosome consists of two sister chromatids held together at the centromere. The spindle fibers, which have been attached to the kinetochores at each centromere, now begin to contract and pull with tremendous force. The sister chromatids, which are identical copies of the same chromosome, are about to separate. This separation marks the key event of anaphase. Now the separation begins. The centromeres divide and the spindle fibers contract, pulling the sister chromatids apart toward opposite poles of the cell. Once the sister chromatids separate, each chromatid is now considered an individual chromosome. The cell now has twice as many individual chromosomes as it started with. This movement continues until each group of chromosomes reaches the opposite poles of the cell. The spindle fibers ensure that each future daughter cell will receive exactly one copy of every chromosome. Anaphase is complete when all chromosomes have successfully moved to their respective poles. The cell is now ready for the final phase of mitosis, where it will physically divide into two separate daughter cells. We now enter telophase, the final stage of mitosis. The chromosomes have successfully separated and arrived at opposite ends of the cell. During telophase, three important events occur simultaneously. First, nuclear envelopes begin to reform around each set of chromosomes at opposite poles of the cell. The nuclear envelope, which disappeared during prophase, now reforms around each group of chromosomes. This creates two distinct nuclei within the same cell. Second, the chromosomes begin to decondense. They unwind and spread out, returning to their less compact chromatin form that existed during interphase. Now, cytokinesis begins. This is the process where the cytoplasm divides, physically separating the cell into two distinct cells. The cell membrane begins to pinch inward from opposite sides. A contractile ring of proteins forms around the cell's equator, gradually constricting like a drawstring. The result is two separate daughter cells, each containing an identical copy of the original cell's genetic material. These cells are genetically identical to each other and to the parent cell that began mitosis. Mitosis and cytokinesis are now complete. Each daughter cell will enter interphase and begin its own cell cycle, potentially dividing again when the organism needs more cells for growth or repair.